Namaskar dosto, welcome to our YouTube channel StudyWire. Myself Vikas Kauda. Today we are going to discuss about cell division. Cell division is an important process in all living organism. Growth and reproduction of cell are the characteristics of every cell, indeed of living organisms. All cell gives reproduce to another cell by giving the reproduced cell is called parental cell, and newly reproduced cell is daughter cell. Every daughter cell going to reproduce the another daughter cells. Like that, the populations of cells increased and growth of living organism can be expected here. In other words, such cycles of growth and division allow a single cell to form a structure consisting of million of cells. And let's start our cell division journey. And just start our cell division journey. In cell division, there are two parts. One is karyokinesis, another one is cytokinesis. Karyokinesis means division of nucleus and cytokinesis means division of cytoplasm. Here two phases are involved in cell division. In karyokinesis there are two phases. First one is interphase and division phase. Interphase is the longest phase and division phase is shortest phase. Again, interphase is divided into G1 phase, G2 phase, S phase, and division phase is divided into prophase, metaphase, anaphase, telophase. Division phase is nothing but mitosis or meiosis. Now, going to cytokinesis. In cytokinesis, it is the end product of telophase. The division of cytoplasm is occurred in end cycle of telophase, and then cell division is mainly divided into mitosis and meiosis. Mitosis is occurred in somatic cell. And meiosis is occurred in reproductive cell. Mitosis is followed by meiosis. Again, mitosis is observed by Strasberg in 1875 in plant cells, and in animal cell Fleming observed in 1879. Again, in meiosis, Van Blender observed the meiosis process in roundworm in 1863, and the term meiosis is given by Farmer and Moore. Let's move to the next phase, that is interphase. As we know, cell going to reproduce another cell, then again daughter cell reproduce to their daughter cells. This cycle goes on continuous. In this cycle, interphase plays major role. Here in interphase there are three phases are there. One is G1 phase, another one is S phase, and another one is G2 phase. After reaching all these phases, the cell enters to M phase, that is mitosis phase or meiosis phase. First let's discuss about G1 phase. G1 phase is very short phase, and here no synthesis of DNA or no multiplication of cells takes place in this phase, and it is very short phase, and even in some cases it may absent also. Dividing cells are permanently absent here. Enzymes for DNA synthesis are synthesized in G1 phase. Then cell attains continuous growth, but as I told, in this phase there is no multiplication or synthesis of cell in this G1 phase. Then after reaching this G1 phase, cell moves to S phase. In S phase, DNA synthesis occurs and all histones are synthesized in this phase. Here DNA number is increased but chromosome number is same. In S phase, DNA number increases but chromosome number remains same. For example, let us take a diploid organism present in G1 phase. After moving to S phase, the number of cells increases, and in S phase it remains same, that is diploid condition only. This is its meaning. The chromosome number is same but DNA number increases because synthesis of DNA takes place in S phase, and DNA replication also takes place in nucleus, but centriole duplicates in cytoplasm. This is the main key feature of S phase. Next, after reaching S phase, the cells move on to G2 phase. Here, protein synthesis takes place. After synthesis of DNA, protein synthesis takes place in G2 phase, and here cell is metabolically very active. And after attaining G2 phase, cell goes to G0 phase, that is resting phase. Here cell metabolically activate but it does not proliferate. Proliferate means it does not increase its. Let's see here. As we discussed, cell undergo first G1 phase, then S phase, then G2 phase, then it goes to M phase, that is divisional phase. The cell repeats throughout. In S phase, DNA synthesis takes place. In divisional phase, this may be mitotic phase or meiotic phase. In G1 phase, here cells are metabolically active. In G2 phase, cells are metabolically active and protein synthesis also takes place here. These are all the consequences of interphase and cell cycle. Let's move to next phase, that is M phase, that is divisional phase. Okay guys, next let's move on to divisional phase. In divisional phase there are two phases are there, mitotic phase and meiotic phase. Okay guys, let's move on to the divisional phase. In division phase there are two phases are there, mitotic phase and meiotic phase. In each phases, the phases are divided into four sub phases, that is prophase, metaphase, anaphase, telophase. Let's remember PMAT. This is the code word PMAT. Prophase, metaphase, anaphase, telophase. Now let's study about mitotic phase. Mitotic phase is also called as equational cell division. Let's conclude in the end of conclusion why mitosis is called as equational division. Come, let's study about mitosis phase. As we know about mitosis is only occurs in somatic cells. First one, the first subphase of mitosis is prophase. Here, chromosome become thicker and condensed. In prophase, chromatin is visible. This is the characteristic feature of prophase, and spindle fiber is not yet formed. Simply the fresh cell which is going to divide, it is the characteristics of prophase. And the second subdivision phase is metaphase. Here there are four characteristic features. These are all four characteristic features which is very important for your examinations. First characteristic feature is disappearance of nucleolus, and second characteristic is breakdown of nuclear envelope. You know, the nuclear envelope and nuclear membrane, these are breakdown. And third characteristic is appearance of spindle fiber. Spindle fiber is appeared from centromere, and equatorial plate formation is appears in metaphase. This is main characteristic feature of metaphase. All chromosomes lies center of cell. This lying of center is termed as equatorial plate or metaphase plate. These are all the characteristics of metaphase. Okay, let's move on to next phase, that is anaphase. Anaphase is the shortest subphase and prophase is the longest subphase of M phase or mitotic phase or meiotic phase. Here in anaphase, centromeres move towards the opposite pole of cell, and in animal cell, the formation of spindle fiber is by centrosome. Here centrosome have gamma tubulin. This protein helps to formation of spindle formation. And in plant cell, microtubule organization center helps to formation of spindle fiber, and the cell next undergoes to telophase. In telophase, sister chromatids are reached to opposite phase and nucleolus reappears. Here blast reappears at equatorial plate when it is in M phase, that is phragmoplast reappears at equatorial plate when it is in metaphase. Phragmoplast is nothing but complex arrangement of protein which helps to cell division. These are all the phragmoplast. Phragmoplast appears at the equatorial plate, and as we discussed earlier, cytokinesis is the end product of telophase. In telophase, the nucleolus is reappears and cell cytokinesis occurs, which leads to daughter cell formation. I discussed mitosis is equational division. Why the equational division means when the cell undergoes prophase, then metaphase, anaphase, telophase, it gives two daughter cells which are genetically similar, and one cell divides to two cells, which is equational division. That's why it is called as equational cell division. And let's mention the significance of mitosis. Mitosis is helpful in cell repair, and whenever the wound happens, it repair the cells and synthesize the new cells there. And next significance is meristematic tissue. In plants, meristematic tissues undergo mitotic division, which leads to continuous growth of plants. And the daughter cells are clones to each other. These are all the significance of mitosis, and this video is completed here, and meiosis is coming in next part. So thank you to one and all.